Hey biology students, this is the second video for molecular genetics, and the title of this video is DNA and RNA. So in the previous lecture video, we described the structure of the DNA molecule, and in this comparison chart, we are going to compare DNA to RNA, which is another type of nucleic acid, and then we'll also compare that to protein. So in the previous lecture video, we learned that DNA, the structure of DNA is described as double-stranded. So there are two strands of nucleotides that bind together by hydrogen bonds at the complementary base pairs. So recall there is a four-letter alphabet of the DNA molecule. That is a chemical language, and we have these four letters that represent the chemical letters, the chemical language of the DNA molecule. Let's just write in those letters as A, T, C, and G, although you should know their full name. So adenine, thymine, cytosine, and guanine. You should also realize that DNA gets its name for a sugar, the sugar that makes up the sugar phosphate backbone of DNA. So the sugar phosphate backbone refers to the sides of the DNA molecule, and those are composed of deoxyribose. And this is, of course, where DNA gets its name. DNA stands for deoxyribonucleic acid. And that's what DNA stands for. And that has to do with the sugar. So in this figure, we'll go back in a minute, but in this figure, they're showing us deoxyribose sugar looks like. It is a 5-carbon sugar. However, if we compare it with ribose sugar, it has an oxygen removed. You see that? So ribose has an oxygen here in red, but that oxygen is lost in the deoxyribose, and deoxy just means to lose an oxygen. So it actually is missing an oxygen here and just has a hydrogen. And that's the only chemical difference between deoxyribose and ribose sugar. Both are 5-carbon sugars, or what we call pentose. We would call that a pentose sugar. And these make up the sugar phosphate backbone, or the sides of the DNA molecule. Now, I also left in these numbers that correspond to the labeling of the carbons. So there's 5 carbons involved in this 5-carbon pentose sugar, so we number them. And that becomes important in the orientation of how we write out the DNA molecule, because the 5th carbon is attached to, when it makes a nucleotide, remember the 5th carbon is attached to phosphate, and when that is oriented with the 5th carbon attached to phosphate, that's called the 5' end. And the 3' end is when it's oriented so that the 3rd carbon is pointed in a direction, opposite direction as the 5' end. 3' end would be in the opposite direction. So this would be what we call the 3' end, which is a little apostrophe next to the 3 or next to the 5. Notice here on ribose sugar, we have the same numbering of the carbons. The only difference, again, being there's an oxygen in ribose, and there's missing an oxygen in the deoxyribose. So going back to DNA, DNA has deoxyribose sugar as a component of the DNA nucleotide. So now let's compare that to RNA. So you can see here the RNA molecule is composed of one strand. It's a single-stranded nucleic acid. And I bet you can guess what RNA stands for. So if DNA stands for deoxyribonucleic acid, what does RNA stand for? That's right, it would stand for ribonucleic acid, named after its sugar. So ribose sugar make up the sugar phosphate backbone. Now let's take a look at the alphabet of RNA, the chemical language. So in the diagram, we see that there are 3 nucleotides that are in common with DNA, and that would be adenine, cytosine, and guanine. So those 3 are conserved, meaning they're also found in DNA. The only difference being the ribose sugar making up that nucleotide rather than deoxyribose in the DNA nucleotide. So we would still refer to the monomer. So we refer to what is the monomer for RNA? We would call it a ribonucleotide, which means that it's composed of the 3 things, which would be phosphate, sugar, and a nitrogenous base. Now, notice though that thiamine, the T base, is missing. And in its place, we see a new base. This is called uracil, or labeled with a U. So let's write that in actually with a different color just to show that is a unique base to RNA. And that's a really important thing to notice. So U, which stands for uracil. So in RNA, there is no thiamine. Uracil looks a lot like thiamine. Okay, but that is the main difference. Let's talk about here real quick. Monomer for DNA. The monomer would be this, deoxyribonucleotide. That's just to differentiate between the DNA nucleotide and the RNA nucleotide. Now pictured here, I also have protein. So recall protein. This is going to be important in our next video, where we talk about how we are made out of proteins, and of course the instructions for building proteins come from your genes. They come from your DNA. So proteins are different than the 2 types of nucleic acids pictured here, because proteins are 3-dimensional. And recall we use this word polypeptide. So proteins are 3-dimensional polypeptides. What are they composed of? Amino acids. Okay, so they're composed of amino acids, and there's no sugars. No sugars there. So different, entirely different type of molecule, but has a 3-dimensional shape to it, and that is because we are 3-dimensional. When you're looking in the mirror, you're not seeing DNA or RNA, you're seeing proteins, the shape of your nose, the color of your skin, your hair, all of that, that is protein. So that is a product of your genes. Also want to show you this type of RNA is important. So there's actually 2 types of RNA that we learn about as we discuss how DNA is used by cells to decode our genes into proteins. Well, there's 2 types of RNA that are important to know. So one of those types of RNA is pictured on this slide. It's called tRNA. tRNA stands for transfer RNA. What does it transfer? Well, it transfers an amino acid. So there's an amino acid that's attached at the 3' end. So this transfer RNA transfers amino acids to the ribosome. You recall the ribosome is the organelle where proteins are made. Okay, transfers amino acids to the ribosome during protein synthesis. So when proteins are synthesized or made in the cell. That's the job of transfer RNA. Now let's talk about the second important type of RNA. And that type of RNA is called messenger RNA. And we usually write that as mRNA, which stands for messenger RNA. So this is the second type of really important RNA that's used in the cell. And the messenger RNA acts as a temporary copy of the DNA. Think like a photocopy, okay? Temporary copy of the DNA or a photocopy of the DNA. So in the previous video, we described how we would write out the DNA molecule if we were going to write out the sequence of deoxynucleotides. So let's come up with a sequence here. Remember, we label one side 5' and then the opposite end would be 3'. And then, of course, DNA is double-stranded, so there would be a complementary strand to this DNA molecule. So we would write that with the anti-parallel strand. You should be able to now write that complementary DNA strand based on the DNA base pair rules that we learned about in the first video. So this process is called transcription. This is also a process that is catalyzed by a specific enzyme. And the purpose of this process, this copies DNA. And we're looking here at a specific gene. So usually it's a specific gene. In the previous unit, or previous week, we talked about genetics and how we have genes that code for your physical traits, like your eye color, hair color, the blood type that you have, all sorts of physical care and physiological traits are coded for by your genes, and genes are just segments of DNA. So in this process of decoding the DNA, one step is called transcription. And that word transcription has this word script in it, which means to copy, to make a copy. So this is a process that copies DNA, a specific gene, into mRNA. So it makes a copy of DNA into messenger RNA. So this is going to be catalyzed by an enzyme. And you need to know the name of this enzyme. This enzyme, recall in the previous video, we learned that DNA is copied into DNA during DNA replication, and that enzyme is called DNA polymerase. So in this video, we're learning that DNA is copied into mRNA when the cell wants to create a specific protein. The first step is transcription. So the DNA is copied into mRNA, and that enzyme is called RNA polymerase. Isn't that nice? So the name of this enzyme, we know it's an enzyme, an end in ACE, and enzymes oftentimes tell you what they do. They tell you their job. So their job is to make an RNA polymer. So it's going to copy the DNA in order to make an RNA polymer. So in order to do that, we have to remember what we just learned, that in the RNA molecule, there is no thymine. There is no T base. So in place of T, there is U. So U would be found in the RNA, okay, where the T is found in DNA. So the concept here is that U replaces the T. Okay, so let's do our copying of the DNA. Now, in the copying step, there's one strand, and it's this bottom strand here of the DNA that we refer to as the template. That means this is the strand we're going to copy from. So we're going to take that template strand and we're going to write it, we're going to transcribe it, we're going to copy it into RNA. So RNA polymerase will have a 5' end here as we begin copying our RNA polymer, our RNA transcript. And so notice now, if we have a T in the DNA, we can add an adenine in the RNA. Okay, so we still use those same base pair rules, and the C pairing with the G, that is still the same. But now here, when we see an A in the DNA, so this A in the DNA, rather than putting a T in the RNA, we would be putting a U. So there would be a U there in place of the T. Okay, so we go and keep copying. So in this example, we have this U here instead of thymine. Notice something else. So one way to transcribe this DNA molecule is you can look at the template strand and then you can write the complementary strand. But you can also notice that this mRNA has the same sequence as the DNA gene, with the only difference being that there is a U instead of T at this particular point here. And that's because this strand of the DNA molecule, we refer to that as the coding strand. So the coding strand has the code that is going to be transcribed. You know, that's the exact sequence. It's the same as the coding strand of the DNA, except now we have uracil instead of thymine. But it's the complement of the template strand. Okay? So in the next video, we're going to describe how transcription fits into a process that we call gene expression, or the central dogma of biology. So, talk to you next time. Bye.